18 multiple choice for the hire. Okay, this is the 2018 multiple choice for the hire. Okay, following substances are products of fermentation. We have ATP, lactate and carbon dioxide. Which of these are products of fermentation in human muscle cells? Okay, so human means that we're looking at animal. And so as far as animal fermentation goes, we get our first stage, glycolysis still occurs, gets you to pyruvate, which then straight away is converted into lactate, nicely reversible. This is a nice example of a reversible reaction. You might want to use that for metabolic pathways. But during glycolysis, this bit for your glucose to pyruvate, you have your energy investment stage where you put in 2 ATP and get out 2 ADP and PI. But you actually also generate enough to get 4 ATP, so that's your net gain in the energy payoff stage. So what do we get? We'll get some ATP, although only a pitiful 2, and we'll get lactate as well. Okay. B. Okay, question 2. Diagram represents a stage of cellular respiration that occurs in a mitochondria. Okay, you're told it's the inner membrane proteins. This should automatically be telling you that you're looking at, oh sorry, change this back. You're looking at the electron transport chain. Okay, so this is, oh, well that doesn't make any sense. Let me just get rid of that. So what we have here is electrons, high energy electrons that are being used by protein pumps to power the movement of hydrogen ions into the gap between the two membranes of the mitochondria. So dodgy drawing of a mitochondria, but we get the doodle. That obviously should be closed. So our little pumps are on here, which are pumping in hydrogen ions, which then come back through on their gradient now through ATP synthase, which would be in here, and that makes ATP. Okay, so the final, let's work back the way actually, I think is easier. So the final one, to make water, we need something that can accept the high energy electrons and the hydrogen ions at the end. So this here is oxygen. So what we need here is our hydrogen ions and our electrons, which also are here, because it's the same letter. Okay, these have been dropped off by NADH, which has dropped these off here, and it has gone back to NAD, and that will now go back and pick up more of them. Okay, and the whole point of this entire thing is to make ATP. Okay, so our final answer is D. Oh, question 3. Oh, I can't seem to get it just right. I might need to move it up and down. Part of a metabolic pathway used by cells to produce the amino acid alanine is shown. Okay, so we have phosphoenol pyruvate. Pyruvate kinase is the enzyme that changes that to pyruvate, and then we are not even naming it, but another enzyme must be changing it to alanine. Okay, so alanine is a non-competitive feedback inhibitor of the enzyme pyruvate kinase. So this is using feedback inhibition here. Okay. Following statements refer to the pathway. So pyruvate kinase reduces the activation energy needed to convert phosphoenol pyruvate into pyruvate. Okay, so this is an enzyme. What enzymes do, if this is a normal pathway, is enzymes reduce the bump that you need to go through to make it work. Okay, we have a little EA there. So yeah, that's absolutely true. It has to be there. Okay. For question 2, phosphoenol pyruvate is the substrate for pyruvate kinase. Substrate, enzyme, product. Yep. Alanine combined to the active site of pyruvate kinase. Tells you here, non-competitive. If it's non-competitive, it binds to a site other than the active site. So that is wrong. So it's 2 and 3. Sorry, 1 and 2 only. Okay, not 3. So A. Question 4. Shrews are small mammals. The graph shows a relationship between body mass and oxygen consumption of shrews at 2 environmental temperatures. Okay, here we've got a 10 degree line, which is your solid here, and we've got a 20 degree line, which is dotted. So we can see that categorically the oxygen consumption is decreasing with temperature and is also decreasing with body mass. Okay, Which of the following statements about the correct graph is correct? Shrews of greater mass consume less oxygen. Okay, right, now this is sneaky. This is very sneaky. Okay, this is body mass in grams. This is the oxygen consumption in grams of body mass, so per gram. So if I look at this 20 gram shrew over here, okay, although it's looks, we'll just look at the 20 one because it's lower, okay, this one here is what, 3 point, no, where we're going, so that's 2, so going up in 0.2s, so that is, sorry, going up in 0.4s, so that would be 3.6, okay, centimeters cubed per gram, and it's 20 grams, so we'd have to times that by 20 to work out how much they consume. If I look at something on the other side, so let's look at the 4 gram body mass one, the one over here. Now that looks like it would be bigger. Here we are up at 9.6 centimeters cubed, but that's per gram, and that would only be times by 4. So this number is going to be much bigger than that number. So shrews of greater mass consume less oxygen is not true, but I can totally see how people would get tangled up in that one, okay? The optimum temperature for oxygen consumption was 10 degrees. It's better at 10 than it is at 20, but I have no information about optimum. For an optimum, I would have to have something that showed that, say, 9 degrees was not any better, and 11 degrees was not any better. You would have to have more information than this, okay? So no information here. As environmental temperature increased, oxygen consumption decreased. Well, that is clear. Yes. Okay. And just to check, at 10 degrees C, a 16 gram shrew consumed 6.2 centimeters cubed of oxygen per gram body mass per hour. This is just checking that you've done the correct scale point here. So we were going 16 gram shrew at 10 degrees. Here we go. So it's not, am I on the right place? 10 degrees C. 10 degrees C, that one up there, sorry. 10 degrees C for your 16 gram is actually 6.8, okay, rather than 6.2, okay, because it's going up in 0.4s. So, no. Apologies for the slight scribbliness on that one. Okay, question 5. Yeast cells contain the enzyme catalase, which breaks down hydrogen peroxide to produce oxygen. An experiment was carried out into the effect of lead nitrate concentration on the activity of catalase. Really common setup, this one. Okay, 6 flasks were set up. Each contains 25 centimeters cubed of hydrogen peroxide and 10 centimeters cubed of yeast suspension. 10 centimeters cubed of a different concentration of lead nitrate was then added to each flask. The volume of oxygen produced after 15 minutes was measured. Okay, identify the independent variable in this experiment. Okay, so independent. The independent variable is the one that you control and change. Okay, it is the changing but controlled. Okay, it is the thing that you're really doing the experiment on. Okay, so in this case we're looking at lead nitrate concentration, so that is our independent variable. Just to be absolutely clear, our dependent variable is the thing that you measure. Okay, so independent is the concentration of lead nitrate. This would be the dependent over here. Volume of oxygen. Question 6. Diagram below shows the circulatory system of a fish. Arrows show the direction of blood flow. Which row in the table describes the type of circulatory system of a fish and the blood pressure in the capillaries in the gills and body tissues? Okay, right, so the fish is the simplest system that we can have. Okay, what we have for a fish heart is really very, very simple. Okay, we have an atrium and we have a ventricle, and that's it. Okay, so what happens is we go up to the gills, so we pump it out from the ventricle at high pressure, but high, we'll put BP just to make that work. Okay, it then goes through the gills, and the gills are all millions and millions and millions of different blood capillaries, so therefore the blood pressure will drop. Okay, and then it joins back up again to make the next kind of run to the body tissues, and goes through another whole pile of capillaries. So our blood pressure is still pretty low, in fact lower, and then finally joins back up and goes back into the atrium. Okay, so we are looking for a single circulatory system. Okay, and then we're looking for the difference between the blood pressure in the gills and the blood pressure in the body tissues. Well, the blood pressure in the gills is still going to have that surge coming from the ventricle, so it's going to be higher, and then we're going to end up lower. Okay, so C. Okay, question 7. In an investigation into fermentation, yeast was grown in a flask of glucose solution for 20 hours at 20 degrees C. The graph shows the concentrations of ethanol and glucose in the flask over the period of the investigation. Okay, so higher graphs now, so we're expecting a double axis. So here's our ethanol, that's the solid line, we are reading it to the left. Here is our glucose, it's the dotted line, and we are reading to the right. Okay. What was the glucose concentration when the ethanol concentration was 3.3 grams per litre? So ethanol we're reading to the left, find 3.3. So this is going up in 0.1, so here's my 3.3. I need to read it across to my solid line because that's my ethanol one. Here's my 3.3. Okay, so that's at 17.5 hours. Okay, so go down the way to find out what glucose was at that point. So there's the point, and then read it across the way, and then we just have to be a little bit careful that this one is reading, because there's 3. Okay, so this is reading up in 0.2s, so this one is 2.4. Okay, right, question 8. You'll notice I've pulled in a picture here for this one. The following statements describe stages in the Calvin cycle, so carbon fixation. So I've actually just pulled out a screenshot of one of the slides from the whole photosynthesis presentation. But I would expect you to be able to pretty much do this from memory, because that's what it's asking you to do. Okay, so we've got carbon dioxide attaches to ribulose bisphosphate, producing 3-phosphoglycerate, or 3PG. Okay, 3-phosphoglycerate, or 3PG, forms glyceraldehyde 3-phosphate, G3P, and then your G3P regenerates ribulose bisphosphate, RUBP. Okay, so we've got RUBP going to 3PG. Okay, so this is number 1 happening there on this one. Number 2 is where we have the conversion between 2PG and 3G3P. Always difficult. Okay, and then we've got this coming back the way for the regeneration of ribulose bisphosphate. Okay, which row in the table identifies the stage which is catalyzed by Rubisco and the stage which requires hydrogen? So here is our Rubisco. Rubisco stands for ribulose bisphosphate carboxylase oxygenase, so it has to be the one that is connected to ribulose bisphosphate. Okay, and that's there, so number 1. And then requires hydrogen, you just need to know where that comes in. So the hydrogen is coming in as the NADPH, and that is being dropped off in the stage where we're converting 3PG to G3P. Okay, so where we're going, stage 2 on that one. Okay, I'm afraid you have to know it, so if you have to spend the time, spend the time. Okay, you could write it much simpler. We're going but you need to know the whole thing anyway. You need to know that you've got Rubisco in here, you need to know that we are using ATP in here, and you need to know that we are also using the NADPH transport in here. Okay, so none of it easy, to be honest, and you also need to know that you need a little bit more energy to make that function. And then you need to know that you're pulling off these to make glucose. Find a way, make sure you're happy with it. Right, question 9. Diagram shows apparatus used in an investigation to measure the rate of photosynthesis at different wavelengths of light. Coloured filters were used to change the wavelength of the light. The volume of oxygen collected after 30 minutes was used to measure the rate of photosynthesis. Okay, so we've got a little setup here, that's fair enough. Suggested improvements were: use a measuring cylinder with a narrower diameter, repeat the experiment several times and take an average, use a scale with more divisions. Okay, you're then asked, which of these suggestions would improve the accuracy of the results? So the accuracy of the results has to be about your measurement, not about whether you trust the measurement, because that would be reliability, but how you're actually doing the measurement and how can you make the measurement more accurate in terms of smaller divisions, basically. Okay, so the obvious one that is definitely right is I could use a scale with more divisions. So obviously I can only measure this one. I'm going to have to make a guess. I'm going to say, well, it's that division there. But if I had like another 4 divisions in here, I would know exactly where it is, and that would help. That would be more accurate. Okay, what it would also do is if I made the measuring cylinder narrower, because if I make it narrower, that's pretty much the same as, so if I had these divisions on here, oh, that's not clear, wait a minute, sorry, let me just scribble that out. Okay, if I have a measuring cylinder and I have this is the divisions that I can manage. Okay, and so say each of these divisions is 1 centimeter cubed. Okay, if I then use a narrower measuring cylinder and the divisions are the same distance apart, they're not going to be a centimetre cubed this time. They're maybe going to be like 0.5 of a centimetre cubed. So a narrower diameter will allow you to have a more accurate reading. Okay, repeating the experiment several times is all about reliability. Okay, it is not about accuracy. So correct answer for that one is B. Okay, 10. Diagram shows a perennial weed found in agricultural land in Scotland. Which feature of this weed indicates that it should be controlled by a systemic herbicide? So a systemic herbicide is one that moves through the entire structure of the plant from wherever it is actually applied. And this is your problem. Okay, what we need is something that if you attach it onto the leaf, whatever toxin it is, then spreads through the whole plant into the roots and into the storage organ, and make sure that this cannot just regrow from killing off the leaves. Okay, so that's your problem. Okay, that's 1 to 10.